I mean, we all learn in early science about A's, T's, G's, and C's, and our genetic code consists of millions and millions of pairs. So these millions and millions of base pairs, there can be one little change. So something's supposed to be an A, and instead in this individual, for numerous reasons, it could be a T. So in this gene, we obviously want that to be what it's supposed to be. So CRISPR, we can create complementary base pairs to find that gene sequence, and then it attaches and then can snip out that little tiny base pair. And then we can either combine the DNA together, or we can insert new DNA and make sure that the DNA is now what we want it to be.
Hello everyone. Today I'm joined by a good colleague, Chloe, who not only is one of the most disciplined and hardworking people I know, but also a beginner scientist with interest in medical genetics and pharmacology. During her undergraduate course in biological science at Lancaster University, she completed the lab work and research in relation to the immunity, genetics, and evolutionary biology. Chloe has concluded her degree with 2 theses, including the use of CRISPR Cas9 technology for treating cystic fibrosis, and completing the degree with first-class degree. She is now complementing her skill set with Masters in Entrepreneurship and Innovation, while leading the regional division of 93% Club dedicated to improving social mobility. Thank you for joining me today, Chloe. I'm really excited for our talk.
Thank you. It's an absolute pleasure to be here.
Perfect. So I would like to start by understanding a little bit of your research during your undergraduate degree in biology, and specifically about the CRISPR technology that we want to demystify a little bit today. So could you tell me more about the research and that you've done, and how did you come across CRISPR technology during your studies?
Yeah, so it all started with my 6,000 word lit review on whether CRISPR would be a viable treatment for cystic fibrosis. So I kind of focused on cystic fibrosis and the current environment around that. The current drugs have increased the life expectancy dramatically in the last 30 or so years, from about 18 to about 42. So obviously that's a crazy improvement, but there's always scope to do better. So I was kind of looking at whether CRISPR would suit cystic fibrosis and whether it could be a viable treatment in future. And obviously I have to compare the ethics, the cost projections, all sorts of things, and kind of come to a conclusion. Obviously it was quite an early conclusion because there's a lot more to do and only certain amount of research out there for now. But it was kind of an early investigation into CRISPR and cystic fibrosis, really. As well, in my general degree, we didn't really focus specifically on CRISPR, but we kind of learned of the natural mechanisms and the genetic principles that CRISPR uses, because mainly it's quite a simple mechanism. So it uses just general concepts of biology that you learn from an early age. So yeah, it was kind of a general background of CRISPR. But from my degree, I gained further insight and interest into just general healthcare and treatments and stuff like that. So I focused and specialised on the role of lysosome-related organelles in the response of C. elegans, which is a model organism, and infected it with Ephecalis, which is a bacteria. So we can see kind of changes in gene expressions and stuff like that. So it was a big focus on genetics, and then how we can translate that to findings in humans, because humans are very similar to C. elegans, which is a worm, which is quite weird. But that was really interesting. So yeah, I just have a general interest in tackling disease and improving people's lives, which obviously CRISPR has the potential to do. So I've just done a lot of research in my degree, but also just outside of my degree as well, just through general interest. So yeah.
So can we dive deep a little bit more into the CRISPR technology? Because I know a little bit about the genetics and based on the book, The Code Breakers, and how the proteins work. But how would the therapy work from the scientific perspective based on your studies and the research?
CRISPR it stands for clustered regularly interspaced palindromic repeats. It's quite a long-winded thing, and the definition doesn't really have much relevance, I'm not going to lie. It's kind of just interesting to know And it's actually not as complicated as it sounds. So, like I said, it's kind of the basic genetic principles that we learn. Obviously, we learn of our A's, P's, C's, and G's in undergrad. So basically, as an overview, CRISPR is kind of like genetic scissors that cuts and sticks DNA. So it's a cut and stick mechanism. So any diseases that we have or any genetic basis of interest or DNA of interest, we can kind of look at that and then cut and stick it using CRISPR. So basic fundamentals of biology where we can analyze genetic principles and code and cut it in ways that we have never done before, really.
I like the phrase that you've mentioned earlier, like the genetic scissors. Is that correct?
Yeah, definitely. I often see it called molecular scissors as well, but I definitely like the genetic scissors because it definitely does just cut the DNA wherever you want it to cut it, wherever, yeah.
How does it work in practice? Like, I'm quite intrigued because you need to, it's a sensitive operation. Like, you need to be really careful. How would that work in practice?
Yeah, so we've seen it in practice in bacteria. So we know of CRISPR due to research in the 80s on E. coli and their immune response to viral infections. So basically, bacteria and viruses are kind of in an intertwined arms naval race where they're consistently competing against each other. So a bacteria will evolve a counter mechanism, and then a virus wants to invade the bacteria. So it evolves another counter mechanism, and they're constantly going in this evolutionary time space. So yeah, we've seen it in practice in bacteria. So basically, bacteria store CRISPR DNA after infection of a virus, and then the Cas9 protein chops the DNA. So we've already seen it in practice in bacteria, and it's used in bacteria. And we've just taken that general concept from bacteria and transformed it into a tool that we can use as humans. So it's already in practice, but we just adapted it to use in humans. So we already know a lot about it. We just need to fine tune it in a sense.
Because you've mentioned earlier that we are able to translate or transfer the knowledge between humans and bacteria, right? If I remember correctly.
Yeah, I think it was a woman called Jennifer Doudna won the Nobel Prize in 2019 in chemistry for transferring the knowledge from bacteria to humans and see how we can edit it. So yeah.
Okay, perfect. And what implications does that make, like for this field in general? The fact that we can test on bacteria and apply to human some genetic treatments. Like you've mentioned that we can do it, or you've specialized and you've researched the cystic fibrosis. But I presume the implications are beyond that, right? As well.
Yeah, I mean, there's massive implications in terms of most diseases. I mean, not even just diseases, but it goes to HIV or cancers and everything. So there's massive implications in most things we can use it. So yeah, I think the fact that we know a lot from the bacteria and we can transfer the knowledge holds a lot of importance. And then again, like I said before about the model organisms, we can always use other organisms as well to transfer knowledge and then to humans as well.
Okay, perfect. I would like to move on a little bit to like the future of this technology. I feel like I have quite a good understanding. We've discussed like what it is and a little bit of the background to research of how it evolved. Where do you see the future of the technology in terms of making it accessible? Do you have any cost projections, findings that are available online? I presume it's quite expensive technology at the moment. Correct me if I'm wrong. But where do you see it how do you see it being adapted in the future?
Yeah, the economics of CRISPR is quite an interesting one. Obviously, we always look at science individually, but science is kind of always intertwined, no matter if we like it or not, especially when it comes to healthcare. I mean, health is money, really. People will always invest in the health and want to pay money towards it. Like I said before, CRISPR is made from these really simple mechanisms taken from bacteria. So in itself, it's relatively inexpensive compared to other genetic engineering methods, especially. But yeah, it is quite expensive in this moment. The cost comes from the regulatory structures involved in it. So obviously we're actually altering people's genetic codes, and that can have massive consequences, whether it's off-target effects or the ethics of altering the germline in future generations. So it's the regulatory frameworks are obviously really serious and lengthy as well. Just like in the wider pharmaceutical industry, all drugs, all kinds of methods and tools and everything have to go through the clinical trials of stages 1 to 4. So that's quite a lengthy process where you have to proof the concept in trials 1 and then use it on human tissues and animals as well, and just simulations, and then to humans in small amounts, and then on to more humans and more humans and more humans. So yeah, it's just a massive process that takes a lot of time. Ideally, we get a tool and then we put it on the market straight away, but in reality, that's just not going to happen. So it takes a lot of money and a lot of time. CRISPR as well, because obviously everyone's genetic code is different. So it's going to be personable to a certain individual. So it's not like we can mass produce paracetamol or a lot of drugs. The drugs that I talked about with cystic fibrosis, you can mass produce them in a sense. With CRISPR, you can't do that as such because it has to be specific to that individual. So obviously, without standardization, it's personable. So that increased the cost as well. So just with these two, I think it costs about 1 to 2 million per CRISPR insertion, which obviously brings about the question of who will pay. So obviously in the UK we have the NHS, so I doubt in that kind of system they would want to fork out that much money, especially in an early tool that hasn't been backed by a lot of evidence, or if it has at that point, there's also a lot of off-target effects that could happen. And obviously in the US, I'm not sure if insurance companies would want to pay that much money just straight up front, if you know what I mean. So yeah, obviously the accessibility is a massive problem, and it's not clear how we'll get around it as such, whether we can combat the regulatory structures involved and kind of alter the pharmaceutical industry so it's a kind of faster process, or whether we deal with the problems of the technology. Obviously, I talked about off-target effects, and it can cause more problems if it's not done right. So we need to make sure it is worth the money. So cystic fibrosis, the treatments that I've already talked about, still cost a lot of money. The current drugs must be about £300,000 for a year's worth just for one individual patient. So obviously, in the long run, if we can alter their DNA so that they don't have any problems, don't need any drugs in future, then if we're looking at a longer term project, then that money will be useful. So we have to kind of look at the accessibility and kind of see how we can manipulate it so we can benefit from it.
Got it. Okay. We have so many questions now. So I will go one by one. I think I want to understand a bit more about economics, then safety, and then still about regulation. So 3 follow-up areas, but I'll start with the economics. So the current cost is clear. It's also not something that can be easily standardized. The way I understand, it's quite customized, and the long-term benefits can outweigh the short-term costs, but really in the long run. You've mentioned research that takes place and how complicated it is to make sure that this technology is safe. Do you believe that this is a big chunk of the cost, that once it's completely researched and proven safe, let's say a treatment for a disease, then the cost should go down? Or because it's so customized and so personalized to everyone's different DNA, the cost will likely always go high, and it will be just a difficulty of prioritizing which disease and which individuals meet certain criteria to get it?
Yeah, no, I think we will be able to get the cost down quite substantially as well. I mean, we say everyone's genetic DNA is different, obviously, but a lot of the time it is the same. Like cystic fibrosis, for another example, is always caused by certain defects in the channel proteins. So there can only be so many defects in a sense. So obviously everyone's genetics are different in the sense you don't know how you're going to react to certain treatments and stuff like that. But there's fundamental similarities that we'll be able to adapt to and make sure CRISPR is adapted to that as well in future. So I definitely think as long as we combat the regulatory structures and stuff like that, then the standardization won't be as much as a problem. So I think in future it'll be okay.
And maybe just to understand a bit more, because I feel like maybe I didn't cover it earlier in as much detail as I wish I did. You've mentioned that, for example, we can use the cystic fibrosis, right, as an example, because that's the well-researched area for you. You've mentioned that it's usually caused by certain defects in the DNA code. Can you explain that a little bit to me?
Yes, so most genetic diseases will, or even HIV infections and stuff like that, it'll be due to specific alterations in the genetic code. So obviously, I mean, we all learn in early science about A's, T's, G's, and C's, and our genetic code consists of millions and millions of pairs. So these millions and millions of base pairs, there can be one little change. So something's supposed to be an A, and instead in this individual, for numerous reasons, it could be a T. So in this gene, we obviously want that to be what it's supposed to be. So CRISPR, we can create complementary base pairs to find that gene sequence, and then it attaches and then can snip out that little tiny base pair. And then we can either combine the DNA together, or we can insert new DNA and make sure that the DNA is now what we want it to be. And obviously, in terms of genetic disease, if genetic disease is caused just by that one little alteration, then the genetic disease is cured in a sense.
That's mind-blowing, actually.
Yeah, it's quite crazy.
I think I understood it now. And then you've mentioned, because there are certain types of the alterations or changes that correspond to certain diseases. That's why we are able to diagnose those diseases, because we can, for example, see, okay, this is a T instead of an A, of course, in a bit more complex combination of alterations and changes. And then a treatment can be sort of standardized, because it basically has to identify those specific alterations and then replace them with something specific, like a healthy DNA. Is that the right term to use?
Yeah, absolutely. I mean, now we're in a position where scientists have been researching on this topic, so we can actually, with any disease or anything that we want, we can program CRISPR using gRNAs, which is the little code essentially that tells CRISPR where to go. So it's the little tiny gRNAs that can tell CRISPR where in the genetic code it wants to go. And we can do that with literally anything that we want to do. So it is quite amazing. Yeah.
That is amazing. But that kind of sparks my second follow-up question to the previous topic about safety. How do you make sure it's safe? I mean, I can see it can go wrong, right? Very quickly. If I didn't know what I was doing with my own DNA, let's say.
The safety obviously is a massive research basis for CRISPR. So a lot of it comes from the clinical trials that we need to do. The first trials will prove that it's just generally safe. Then obviously, as we get into humans, we need to make sure that it does what it's supposed to, for one. So it's altering the specific little base codes that we talked about that we want it to, and it's not snipping any part of the gene that it wants. So yeah, it's through those type of trials that we can make sure that it is safe, and we need to make sure it's safe, obviously. So yeah, but obviously with CRISPR, because we're altering genetic codes, there's obviously going to be longer term things that we need to analyze. So accidentally snip the wrong part of the DNA, but the implications might only come in 10, 15, 20 years or something like that. And there's also a massive research base that looks into how, if we don't focus on the safety, then say if we're trying to cure cystic fibrosis or anything like that, then later on what we've actually done can cause cancer, because cancer is caused by changes in genetic codes. So we need to look into all different avenues of what it can cause, and obviously widen our scope and make sure that it's safe in the long run as well.
Okay, wow. So definitely the clinical studies are going to take some time.
Yeah.
There will be extended monitoring period. If a parent had a genetic treatment, would it affect DNA of their offsprings in the future? And then if yes, is that taken into the safety trial consideration as well?
If we have treatment on the genetic codes as they are, then they will not be passed to the offspring. It's only if we do this kind of thing in embryos and the germline that it will later be passed through to babies. Obviously, that's another whole other subject, but in a sense, if we're treating genetic disease with CRISPR, then that won't have longer term effects, if you know. So we're kind of just looking at the lifespan for now, unless we are working on the germline.
Okay, got it. So in theory, if I treated a disease genetically, that shouldn't necessarily affect in any way the DNA of my kids.
No.
Other than reducing the risk of the genetic disease, of course, being passed on.
Yeah, definitely. Yeah, you've got it right.
Okay, got it. Fascinating. And I guess the third follow-up was around the regulation and a bit more to understand a bit more what's going on in the regulation space, right? Because a couple of examples that I could read in the book is the technology is expensive. Like you've mentioned, $1 to $2 million. There are a couple of billionaires who might just want to have fun with the technology, right? I want my kids to have, I don't know, great tennis skills or blue eyes or like whatever reason. I guess first of all is how are those claims valid? Is that possible to do or engineer? And then the second of all is how is that technology regulated to make sure that it's used and accessible to the people in need?
Yeah, so you are right. CRISPR does open up a massive wormhole of ethical debate. There's loads of things out there if anyone's interested to look at ethics. I mean, a lot of it applies to just general genetic engineering methods. A lot of it is the same, but obviously CRISPR comes under that thing where we can kind of do what we want, or if it gets into the wrong hands, we can create more harm than good in a sense. But in terms of accessibility, obviously there is the potential for a kind of dystopian future where only the wealthy can afford treatments, which is quite a perverse, but might be a realistic considering pressures from producers and the pharmaceutical industry to kind of get return on that investment. And obviously they've put a lot of energy, a lot of time, a lot of money into developing this product. They want to make money. So obviously they might go private and just get whatever money they can from. So yeah, it's quite an interesting topic. Obviously in today's society, we want a fair society where everyone has access to healthcare in the same way. That is the fundamentals of what everything, especially in this country, the NHS is based on. So I think we definitely need to consider that. The regulations at the minute surrounding accessibility and stuff like that, it's not addressed as such, but there's just general regulation surrounding CRISPR. But I'm sure the accessibility part we'll get into as it comes to market or is closer to coming to market. Well, currently, CRISPR is regulated under the FDA's 1988 guidance on human somatic and stem cell therapy, which obviously 1988 is a long time ago and it probably needs to be updated. And it's kind of put into the same realm as all the other genetic engineering methods, which even though it is similar, CRISPR can be a lot faster, it's a lot more specific. So I think we do need to focus on regulation a lot more. There was actually the Jennifer Doudna who won the Nobel Prize for adapting CRISPR to humans, actually called a moratorium. So she basically said that we need to stop using CRISPR and researching on it in certain ways until we know more about the ethics and how we can do it sustainably and as safely as possible. So she even called for more regulation and to enhance our understanding before we get into these wormholes where we create more harm than good. So that is quite interesting and shows that these are really serious topics to consider.
Okay, perfect. Thank you so much. So I guess as the last question, I would like to ask you about how it affects and how non-scientists can get involved. I think there is a clear interest and a commercial opportunity for scientists, government, of course, patients, and pharma companies. But if I was a non-scientific person that I want to somehow get involved in this technology or I want to educate myself, what would be your advice? How I can get involved in the CRISPR technology area, whether by educating myself, participating in studies, volunteering, what a member of a society can do?
I absolutely love the topic of getting people into science, so I do like this question. I really believe that science is for everyone. So, as many people we can get into science. If you're not a traditional scientist, then the better. In terms of clinical trials and stuff, if you're someone with a genetic disease or know someone who has one, it is very likely, as I think it's 10% of the UK population has a genetic disease. So obviously that's quite likely that you'll know someone or have one or whatever. So there'll be more and more clinical trials coming out at the minute. But obviously in the early phases. So I think there's one for diabetes that has come out in 2022, but I think it only has 8 participants. So for now, clinical trial involvement is quite rare. But I think as we progress through the years and we get further into the trials, then the opportunity for people to get involved in those clinical trials will be more and more.
So thanks a lot, Chloe. That was amazing chat, and then we'll talk soon.